All right. Good morning. Good morning. Welcome to our small group session for today. It's our Tuesday small group session in advance of our interim this week. As some of the little things change in the course, we've consistently had an async video with an all-in since the second week of school. But this week we do not have an all-in, and instead we have the interim. But we do have an async video that kind of covers where we're going to be at this week, so we're still moving and progressing through the lesson. So that's over the cell cycle and cell division. We've moved from the very beginning, opening concepts of biology, the study of life. We've looked at it from a micro, kind of going out to a macro level. In this semester so far, we've covered a lot. We're a little over halfway through the semester, uh, and and in doing so, we look for ways to kind of assess and see where we're at, and that's where our interim comes in. Your interim is taken from released EOCs, so the last few biology EOCs that we've had. We take questions from that and build a 20 question interim. Maybe I think it's 20 or 22 questions. I can't remember, but I know we're in that 2022 range. But they are questions that were directly taken from the prior EOCs. And I thought today would be a great opportunity in advance of that interim, which is due tomorrow, but it's opened up today, but you do have some time to take it as well, is kind of doing a review. And I thought the small group session would be a great opportunity to do that. So we're going to be looking at some of the concepts that have framed where we're at in biology now, and kind of help us better understand and prepare us for that interim as well. That we're going to be moving, because I know we do have several students that are in the session and have been in our session that were late starts. So it gives us, I know we provided all those resources to kind of get you in the all-ins, but I think it's always nice to see all of it kind of together and put together. So today we're going to be looking at a review for our interim. As with all of the classes, I try to give us multiple options and multiple ways to experience the class. I've taken this class and I've placed our slides and our activities in Nearpod, so you can experience it in Neuro, consecutively and concurrently in Nearpod as well. So what I'm going to do before we get started, and then I'll be doing some of the activities in Nearpod, bouncing back and forth as I've done often, both Nearpod and in Neuro. I always get those confused when I say them like that. But it's great to see you guys here today. It's great to have you, and we'll talk a little bit about our community, our class, get set with our norms, and then we'll hit our review. Okay, so I do. Let me go ahead and share my screen for the Nearpod. If you want to participate using the Nearpod in all forms of participation or experience the class as part of the Nearpod. Give me just a second. I have all of my screens maximized, so I have to minimize them. That's now, it should let me do it. All right, I'm now sharing my screen. If you can see that, let me know that you guys can see it. Awesome, great. So the link to joining the Nearpod, I'm going to place in the chat. Okay, and then our class code, I'm going to place in as well. I'll give us about 15, 20 seconds to get, well, give us about 30 seconds to get logged in for those that want to experience it through our Nearpod. And I do advise that, as there are some links and things that are available in there. Welcome, welcome. Good morning. Good morning. See, we have a few more that have dropped in. All right. See, we have some that are logged in. Great job. Great job. All right. Now what I'm going to do is I'm going to come back to our, let's stop our desktop share for a minute. We'll talk a little about our norms and procedures, and we'll go ahead and get started. All right. As always, our class norms really represent who we are, and what we stand for in this course. And in this type of review as well, it's really important for the participations that we ask questions. As always, we are respectful of each other, respectful of the tools, whether that is drawing, pointing, chat, comments, over the mic, all of those things where we respect the tools as well as we respect each other inside the class and how we use and how we relate and discuss and talk to each other. I always focus on the positive. There's so much negativity out in the world. Why bring that here? This is our class. This is our community. And in doing so, I encourage you to ask questions, have an open mind, and through that process, we can have fun in the learning process. What I'm going to do is I'm going to authorize and give everybody the ability to draw or to mark on the screen. Let's see if it'll let me. These things are always wonky. Okay, you should all have the ability. Well, no, I think I took it away. Let's try it again. All right. You should all now have the ability to mark and write on the screen. If you see there's a little small green pencil, if you'll click on that pencil, should open up the tools to be able to draw. There we go. And I would love to see you place your initials as I am doing on the screen to show that we are all in. We are ready for our review. We are ready to participate. I'm loving that response already. I see we have a few that have already signed it. Great job, guys. Show that we are ready. I know we have almost a half of the small group in already. I'm loving that participation. I'm loving you guys showing up. Let's go ahead and see if we can't fill up this board with signatures. I love to see that we're all in, that we're participating, that we're available and able to use our tools. Make sure that you are. I see that we are getting there. Awesome. Want to hear from you. Great job, guys. Let's keep on filling that up. I'm going to give us about another 30 seconds. Encourage your classmates inside of the chat to participate. This is all, as I said, through participation and encouragement and asking questions and all those things, we'll have fun in the learning process. You guys are doing a great job. I'm loving seeing those responses. Thank you guys so much. All right, I see that our brains are warmed up. We're ready to go. We have our tools. We know what we're doing. We know how to access certain things in the course. Now it's time for us to, should we put that in there? I just made some things disappear. Give me just a second. All right, great job, guys. All right. So as with all of our sets, as we said, with our relationship to the course, our relationship to our yours is really tied upon the academic vocabulary. We actually went to we were in a training last week while you guys were not here. Marzano's training, really honing in on that academic vocabulary. So I pulled a couple of really key terms that we've had over this stretch in this semester, and I thought they were important that we can identify them because they're going to come back up throughout our review. So I wanted to kind of set the tone through this word search that we're all going to kind of participate on. To locate these words, become familiar with the words and the vocabulary as we kind of locate them throughout this review. They're going to be essential and important to us understanding the concepts that we have and we've built. Okay. So what I'm going to do is I'm going to open up the screen and I'm going to open up the screen for us to find and locate the vocabulary. Now, what I do ask is that we use a single line, that we mark out our line. We don't double line or triple line, and really so we can be able to see the terms. And then I would love to see if I have anyone that would like to read some of the terms that we have on the board. If you can do so by using the raise your hand tool, anybody over the mic or wants to say some of the vocabulary words that we have here or things that they recognize. Okay. Do I have any of those? Okay. Well, let's do this then. If we don't have any that want to say it at the mic, what I would like for you to do, there is There's a pointer with a little star while we have some that are still marking. Mic doesn't work. I understand that. There's a little pointer that should, if you'll click on it outside of the pencil, you should get an arrow. Can I get as many arrows as I can on the board? So on those same tools, there should be like a little arrow that has a starburst on it. Let's get some pointers on there. I see we have quite a few that are already there. Come on the screen. So terms like organelle and prokaryote and eukaryote, chloroplast, mitochondria, homeostasis, active and passive transport, nucleic acid, osmosis, diffusion, plasma membrane. Then we have thymine, adenine, uracil, guanine, mitosis, meiosis, cell cycle, lytic, and lysogenic cycles. What I would like for us to do, I see that we have those triangles, and I can see who is participating, who's not. I would love to see some more of those arrows and triangles on the board. Let's go ahead and give us about another 10 seconds to get as many as we can on there. Uh, let's go, Caden and Ashley and Isabel and Draven. Let me see those arrows, those pointed arrows. Uh, Jordan, let me see those arrows, guys. All you have to do is click on the arrows. Let me see those triangles, and then I want you to kind of point to a vocabulary word that you may not recognize. Oh, let's see. We always typically point to things that we do recognize. Let's see some of those that we don't recognize. Awesome. I see we have a lot around these words: thymine, adenine, cytosine, uracil, and guanine. And the reason why these are our nucleic acid. These are our base pairs for our nucleic acids. And we'll talk a little bit more about nucleic acids as we have previously. I know we haven't really dove our full lessons into DNA and RNA, but we have reviewed them. And they are parts of some of the knowledge that you'll need to know for this semester. So that's a great thing identifying that. Are there any other things? Mitosis and meiosis. We have not covered mitosis and meiosis yet, but we are coming up on those this week. We're looking at cell division. Mitosis is is the type of asexual cellular division that goes through that helps us replace cells in our body. Meiosis is the creation of the cell division that creates our sexual reproductive cells. So in males, that's sperm, and in females, that's eggs. And over the next couple of weeks, we'll see that. I do encourage you to watch the async video over the cell cycle and cell division that was placed in for the day. So go ahead and look that, and you'll be able to get some introduction to that. And we'll talk about we'll try to touch some of it today as well. So great job. You guys let me know a little bit about what we saw and what we did not see and what we need to kind of hone in on. Great job for letting us, and I'll adjust my lesson so that we can really we'll talk about these base pairs as well. Okay, awesome. Great job, guys. Thank you guys for leaning. Your feedback and your responses help me form the class, help us build that. Great job. So I'm going to go ahead and let me freeze this for just a second. Okay, so biology as a whole really describes what? What does biology describe, guys? If you look at the name of the course, biology, what is biology the study of? Living things. Very good. Life. Excellent. That's right. Biology is the study of life. So the characteristics of the experiences that we study in this semester have been the interactions of living things. Those interactions, and over the next semesters, we'll look at how the interactions those organisms interact with each other. Right now, we're looking at the internal things, how things inside of these organisms are living things are interacting. And there are characteristics of things that are alive. What are some of the ways that we can tell that things are living or dead or not alive? What are some of the ways that we look to tell that things are living or not alive? Love to see your responses or hear your responses in the chat. What are some of the ways that we look at and determine if things are living or not alive? A heartbeat. Okay, very good. A heartbeat. They grow. Yeah, awesome. Yeah. So photosynthesis, breathing, they need food. You guys are doing a great job. So I want you to kind of think about that, and I'm going to refocus and turn that. Here we go. I'm going to turn our ones back on, and I want to see those pointers again. If we'll move over here, there are characteristics of life that are inevitable for all living things. Metabolism, that's a term that describes all the chemical reactions that occur in an organism. And our portion. And all living things have metabolism. There are chemical reactions that are keeping them alive. Okay. And some of you guys noticed something like photosynthesis or cellular respiration. Heredity describes how all living things and traits pass traits on. As we know, we are the byproduct of our parents, right? We get half of our genetic material from our mother, half of our genetic material from our father. They're able to pass those genes along. All living things are able to pass those genes along because the basis of our existence really ties upon that DNA. And those characteristics that are passed along through heredity. A response that there's a response to a stimuli. Things that like cold temperatures make you shiver and those type of things. They're responses to stimuli. Homeostasis. It's the one of our big H words. It's one of our major academic vocabulary words. It means it's a balancing of states. So internally and externally, as an organism, living things balance. They react to things through either positive or negative feedback, and creates a balancing of systems. We keep and balance our systems. This is also a process of that homeostasis. That there's a balance between the systems. As we see, we have an internal core temperature as well as a temperature that we set, and it's balancing. The ability to reproduce is also another characteristic of life. As you guys said, inherited traits. Some of and the answer is yes, being able to reproduce. I would love to say that my cell phone here that was a living thing. I could walk away and come back one day and I would have another iPhone because it would reproduce. But that's not the case. It's a non-living thing. It does not reproduce. And things grow and develop. Living things grow and develop. We start as organisms ourselves, as humans. We start out as infants and we grow through multiple stages. And we'll talk a little about that over the week of why you get bigger. It's not necessarily that your cells get bigger, but you have more of them. And they're the processes of growth and development. And then we have levels of cellular organization. And this is the basis of most of the stuff that we've talked about in biology right now, that there are levels of organization. We start out with in biology, we talk about the most basic portion of life is the cell. But we know that cells are made up of other things as well, of atoms and molecules. In just a few minutes, we'll kind of review some of those biomolecules, those 4 macromolecules that are essential. But at the basis of biology, we start at the cell. Cells make up tissues. Tissues make up organs. Organs make up organ systems. And those organ systems make up the organism. And through that, we have the process that says that things that are living have cellular organization. Even if they're single-celled organisms, they're organized in a way, that cell is organized in a way, and that organism that they're alive. Okay. So great job, guys. Now, what I would like to see, is there any feedback? I would love you to use your pointer. Are there any of these characteristics of life that we have issue with, or do we feel like we're going? What I'm going to do is I'm going to put up a quick poll. Let me know, do we feel comfortable with with our characteristics of life? Let me know what your response is. This will be a key thing that we'll continue to do as we move through. I need to see as many responses as possible to let me know that we got it and we're ready to move forward. So yes, our first part of our lessons, great job. You guys are giving me great responses. The and okay, the beginning of this, so said, as our first parts of our course, we really set upon the characteristics of life, defining what was living and how they were living. Then we went through and started breaking down the organization of life, how things are put together and built. That make up the organism. And looking at that, is the cell is the basic unit of life. But what makes up the cell? The cell is made up of molecules, and there are 4 essential biomolecules that we say that make up a cell or make up life. They are carbohydrates, proteins, Lipids and nucleic acids are four macromolecules, and each one of these molecules is also made up of something, and is built upon other things. The single units that they're made up of are monomers, and this kind of goes back to our academic vocabulary as well. Mono is a prefix. What does mono mean? I would love to see some responses in the chat. What does the prefix mono mean? One. So when we talk about a monomer, we're talking about the single unit, or the single unit. Thank you, Dorian. That makes up a molecule. Uh, when we hear the term poly, what does poly mean? Essentially, you're going to say more than one, but essentially it's going to be two, more than two. All right. We have terms other for two, but two or more, very good when we talk about in terms, okay? We have other specific terms for two, but when we talk about the building blocks, two or more would make up poly. So great job, guys. So as atoms form together, they create an element. Elements form molecules. And polymers are also formed by joining several smaller units or molecules. And we call those smaller units monomers. Each one of our macromolecules, each one of the carbohydrates, lipids, proteins, and nucleic acids all contain monomers, and they also make up polymers. So when you think about them in terms of like Legos, and I had a 3D Lego game that I was going to play through, but there was an error in it after the shockwave, after they've changed all of the systems. But if we build Legos, you could build just remarkable shapes with individual pieces or units. So if we think about that monomer as an individual unit, and we start building pieces, they become something, and then we can take those pieces, prefab pieces. Like my son plays with Legos a lot, and let's say the last thing we tried to build was we have a little space station, and the pieces came in multiple, because they were repeating pieces. So they came already put together in different pieces. Those are kind of like the polymers. They're multiple pieces that you can stack together in complete forms. The monomers are those, think of that as an individual Lego. Thousands of Legos. Yeah. So if we think about that, those are the components that make up those our four major macromolecules: carbohydrates, lipids, nucleic acids, and proteins. Now, with carbohydrates, carbohydrates are our sugars. The most important part of that sugar, the most important carbohydrates, they supply energy. Would be glucose, and it is the byproduct of photosynthesis as well, right? So at the simplest form, those monomers that we call for glucose are monosaccharides, or single sugars. Saccharide is another word that we use for sugars. For lipids, which we use for long-term energy storage, we have glycerols, and those lipids are fats and oils, depending on what they are at room temperature. And the lipids are extremely important to all of our lives. I know we hear about lipids in our diet, but but lipids play other important roles inside our body, like insulation and cushioning. But they also insulate and protect nerve endings, as well as serve as long-term energy storage. Our third macromolecule are proteins, and proteins are help build the structures that we that make up each and every one of us. And they're also steroids and enzymes. Enzymes speed up chemical reactions by lowering the activation energy. And as we've had in previous lessons, I go back to really go watch those async videos over each one of those. But the essential components are understanding of what proteins and they have a special class of enzymes that are like a key to a lock. They're very specific, and they speed up chemical reactions, that metabolism that we talk about, all of the chemical reactions that are occurring. The last biomolecule we're going to talk about are nucleic acids, and this is DNA and RNA, and they're specifically tied for genetic material. And as you guys informed me, what you didn't recognize in our vocabulary were some of the terms that describe the base pairs for DNA. So let's look at nucleic acids for a second, because we did have a little bit of trouble. The base shape for DNA is this double helix. It's kind of turned on its side here, but if you flipped it over, it will look like a ladder that has been twisted, right? If you untwist that ladder, you have basically essentially a you have basically essentially a ladder right here. Like you see that? So it's twisted like this. It's kind of like looks like a spring that's been twisted. But if you unravel it and straighten it out, it will look like this, a ladder. And just like a ladder, you have the poles of the ladder, and you have steps or rungs on the ladder, or steps on the ladder. Can you guys join me? Let's take a second, and we're going to use our pointer tool. Let's kind of go around here and let's look at this ladder. Kind of get you guys to join me. I would love to have all of you, Isabel and Draven, Alexis and Jordan, Caden, Holly and Dorian. Let's get all of you guys in here. Let's look at this ladder. I want you guys to point to the steps or the rungs on the ladder. Point to the steps of the ladder. Let's go to the steps of the ladder. Do we notice something about the color code on the steps of the ladder? You're right, Alexis. We notice something about the color code, and are they the same colors? Love to hear some responses in the chat. Are the steps right? No. And the reason why that's very intentional, if we look over here, the reason why these steps are not the same is that they have corresponding matches. So if we look at this sequence of DNA, we see that none of the steps are different colors, but they're only one or two different colors, right? And that is the reason is because we have base pairs. Those terms or that vocabulary that you struggled with earlier, adenine, thymine, cytosine, and guanine, these are the components or base pairs that make up the steps of the ladder. Now, the cool part is we look at them and we separate them, but we usually refer to them by the first letter of their names: adenine A, thymine T, guanine G, and cytosine C. Now, the cool part or the interesting part is that A's and T's, when we talk about DNA, match up, and C's and G's when we talk about DNA matchup. So if we see this sequence of DNA, it is just a measure of A's and T's and C's and G's matching up to really that what gives us those components. If we think about it and look at it as a ladder, you have the poles of your ladder, you have the steps of your ladder, and you have those you have screws that hold it all together. This combination of this three makes up the monomer for nucleic acids. We call the monomer for nucleic acids a nucleotide. It's composed of carbon, hydrogen, oxygen, phosphorus, and nitrogen. And it's a three-part. It has a base pair, it has a 5-carbon sugar, and it has a phosphate group. If we look at it, the sugar makes up the molecules that we say the phosphate group is kind of like the screws that holds it together, and the base pair is each on each side. So these combine to make your ladder, but are really essentially the monomers, as we say, if you think of them like Legos, that make this ladder together. Okay, and the A's, T's, C's, and G's are the base pairs that really represent it. Now, the other nucleic acid is RNA, and it is very similar to DNA. The only difference between RNA and DNA is that as DNA is a double helix, so it has that double ladder shape, RNA is a single helix. It only has one side. It's a single ladder. It's a single helix. And it only has one side of this. The reason for that is it's used for communicating specific traits moving from different parts of the cell. Now, and we'll talk about cell transport and protein synthesis much later in the course. But understanding that DNA and RNA contain genetic material is going to be essential important. Now, the other difference between DNA and RNA is that there is a 5th base pair that's called uracil. Sorry. Uracil replaces adenine, or it replaces it replaces thymine when we look at RNA. So when they bond, instead of A's and T's, it's A's and U's, C's and G's. Okay. I know that was a lot, so let me go ahead. I'm going to put a poll out. See how do we remember and understand what is our comfort level with our biomolecules. Okay. Now we will continue to grow our knowledge of of our nucleic acids, DNA and RNA, over this next couple of weeks because they're really important when we talk about DNA. Uh, when we talk about the replication or understanding of how DNA works with the cell division, but we're moving forward on that. Great job, guys. Go ahead and end that. We're getting through and we'll move through a little bit. As we said, the basic unit of life is in the cell. It contains DNA, which is the blueprint for growth and development. Excellent job, guys. So what is the difference between a prokaryotic and a eukaryotic cell? Now that we know, or we've talked about that the cell is the basic energy, what is the difference between a prokaryotic and a eukaryotic cell? What I'm going to do is I am going to give us about 2 minutes. I am going to put us in a breakout room and I'll let you share whoever you're in the breakout room with. I want you to tell me the difference between a prokaryotic and a eukaryotic cell. And when we come back, we'll talk about the difference and we'll move forward through the knowledge of that. Okay. So give me just a second. I'm going to put you all in a breakout room for about 2 minutes. All right. So I'm going to start the rooms. You should be moved into breakout rooms. I want you to discuss what is the difference between a prokaryotic and a eukaryotic cell. All right. Everyone should be back in the room. Can somebody tell me what is the difference between a prokaryotic and a eukaryotic cell? What is the major difference between prokaryotic and eukaryotic cells? I'd love to see it in the chat. If somebody wants to say something over the mic, let me know. What is the difference between a prokaryotic and a eukaryotic cell? Excellent, Dorian. Eukaryotic cells have a nucleus. Prokaryotic cells are less complex. Eukaryotic cells are more complex, okay? Prokaryotic cells. Okay. Eukaryotic cells are more complex than prokaryotic cells. Prokaryotic cells are simple cells. They're when we talk about the origin of cells, they're probably some of the very first cells that we have. Eukaryotic cells are going to be our complex cells. These are what you're going to be found in your multicellular organisms because they have organelles. These are tiny organ-like structures. Eukaryotic cells are only found in plants, animals, fungi, and insects. They are from multicellular organisms. Well, okay. They have a nucleus. They have DNA is located inside, and DNA inside of a eukaryotic cell is the double helix structure is located in the nucleus of the cell. Prokaryotic cells are simpler. They're found in bacteria and other single-celled organisms. They have no nucleus. The DNA in its cells is in a circular fashion and it's found in the middle or center of the cell, but it is not in a nucleus. The difference between a prokaryotic cell is the most important, probably is going to be the presence or absence of a nucleus. Very good. Eukaryotic has a nucleus, prokaryotic, no nucleus. Excellent. All right, let's see. This picture shows a student using a microscope to study a prepared slide of a single-celled organism. The single-celled organism is classified as a prokaryote based on the absence of what? A cell membrane, B, ribosomes, C, chromosomes, or D, a nucleus. I'm going to put a poll out there for us. Which best answers the question? A single-cell organism can be classified as a prokaryote based on the absence of a what? Loved it. A cell membrane, B, ribosomes, C, chromosomes, or D, nucleus. The absence of what? A prokaryote is described based on the absence of what? All right, I'm seeing overwhelmingly good job based on the absence of a nucleus. Excellent job. So remember, we determine prokaryotes and eukaryotes based on the presence or absence of a nucleus. Prokaryote cells, no nucleus. Very good. Excellent job, guys. So we said one of the big H words or one of our vocabularies is homeostasis. It's the maintenance of internal balance in the cell of an organism, either through positive or negative feedback loops. It causes the cell organelles to maintain a stable internal environment. That is the definition of homeostasis. We do this through the movement or transport of organisms or cells through reproduction. We see that the plants and animal cells have things. Just to kind of add on the difference between a virus is not a prokaryotic or a eukaryotic cell. Remember, they are not living. They do not reproduce on their own. They must have a host cell. So they are not prokaryotic or eukaryotic. They are pathogens. They are not living. Only cells that are prokaryotic and eukaryotic cells. So our plant and animal cells are going to be eukaryotic. We have bacteria that's going to be prokaryotic, but viruses are not living. They're not living. Okay. Sorry about my writing. They're not living. Okay. So the transportation of nutrients occurs through the cell membrane, endoplasmic reticulum, and the vacuoles. The most important thing is to know about the cell membrane. This is what allows things to move in and out of the cell. It may be called the plasma membrane, may be called the phospholipid bilayer because it's made up of phosphates and lipids. Remember, we just talked about lipids. Lipids are fats. What do we know about lipids? Fats and water. If you mix fats with water, they don't mix very well. The same thing, that cell membrane is made up of those. So the external atmosphere creates this layer. The way that they organize creates this bilayer of phosphates and lipids because they move away from the water and move towards the water. And it creates a barrier between what's inside of the cell and what's outside of the cell. We call this barrier the cell membrane or the phospholipid bilayer or the plasma membrane. As we see, cells move things in and out of the cell. If it needs energy, we call that active transport. And active transport requires energy. Passive transport does not require energy. So the common types of passive transport are diffusion, facilitated diffusion, and osmosis. And it describes how things move through that phospholipid bilayer. I encourage you to go back through and watch the videos for active and passive transport. Okay. And just to kind of wrap up what we've covered over the last a few weeks, well, the last couple of weeks, we've really looked at the energy needed. We said that active transport requires energy. That molecule of energy that is required is known as ATP. And the creation of ATP is created through the process of cellular respiration. And the photosynthesis provides us with food or the energy in order to go through that process for plants and creates that glucose that we as organisms need. We are not producers, so therefore we cannot make our own food. Okay. Uh, we can't make our own food. Plants do through the process of taking sunlight, energy, and turning that into glucose. We as organisms are consumers. We can take that glucose or by consuming plants or consuming other producers or other consuming other consumers, and we can turn that into energy or ATP. Now, the important parts to remember about the photosynthesis and cellular respiration formulas is that they're mirror images of each other. Okay. I don't want to get you guys, what are some of uh, the components of the photosynthesis and cellular respiration? If I needed to start photosynthesis, what three things do I need in order for photosynthesis to take place? If I needed to have photosynthesis for a plant to start, what three things would I need for photosynthesis to take place? I would love to see that inside of the chat. What three things do you need for photosynthesis to take place or to react as reactants? Very good. Carbon dioxide, water, and sunlight. Those are the reactants for photosynthesis. What would be the products of the photosynthetic reaction? What are the products? What are the things that come out of? Very good. What are the things that come out of the photosynthesis? What things are produced or the products? Glucose and oxygen. Very good. Now we have the reactants of photosynthesis: carbon dioxide, sunlight, and water. And we have our products, things that are produced, glucose and oxygen. Now, if I told you that the processes of cellular respiration were mirror images, what would be the reactants of cellular respiration? What do you need in order for cellular respiration to take place? If these are if they're mirror images, very good, Dorian. You need glucose and oxygen for cellular respiration to take place. What are the products of cellular respiration? What happens at the end? So if you have glucose and oxygen, what happens what is produced? What are the products of cellular respiration? Very good. Carbon dioxide, water, and energy in the form of ATP. Excellent job, guys. Great job. Now, just to make sure that we have these concepts, where does photosynthesis take place in the plant? Which organelle does photosynthesis take place in? Which organelle? Very good, the chloroplast. Which organelle does cellular respiration take place in? Where does cellular respiration take place? Excellent, the mitochondria. So, photosynthesis takes place in the chloroplast. Cellular respiration takes place in the mitochondria. And the processes are mirror images of each other. The products of one are the reactants of the other. The reactants of one are the products of the other. Excellent job, guys. Great job. So, just to kind of let me know a little bit about you, what do we know about the cell cycle? So, what I'm going to do is I'm going to set a poll. Let me know by the faces. How much do we know about the cell cycle? Do we know anything about the cell cycle? Very little. Okay. Seeing some, we have some middle faces, no frowny faces. So I'm guessing that we've kind of looked. Okay. So all living things are made up of cells, and those cells have cells just like we as organisms have cycles. They have a cycle. They live and they die. In order for us to replace or replenish the things that are damaged in our bodies, as well as in order for us to live, at any given time, these cells are going through various cycles. And I encourage you to go ahead and watch the async video for this week to refresh and encourage yourself to know more about the cell cycle. Now, while the interim doesn't hit very much on the cell cycle, it is what we're moving forward through this week. And I thought it was a great opportunity to kind of set the tone. Tomorrow in our live session, we will be focusing in on the cell cycle, and I will get this out to the other students that we kind of covered today. So I want to thank you guys for your participation. I want to thank you for the time that you've given, and you have a wonderful day. Before we close, let me know a little bit. I'm going to put up the smiley faces. How do we feel about our review? How do we feel about taking our interim? How do we feel about the concepts that we're going to have? Okay. That lets me know you guys giving me smiley faces. I would love to see all of you in here. Let me know how you feel. Okay. So I may put together, I see a few of us with some frowning. I may have to put together, maybe actually going through the Kahoot myself and putting together some things. Don't be scared. You guys are going to do great. You know this. Like I said, we just kind of covered a lot of the concepts. We covered most of the things that you're going to see on side of there. So I feel very confident. You felt confident in it. So you guys have a great day, and I will see you tomorrow. Have a great day. See you very soon.